Today we're looking at mutations. And if you are my son, if you hear mutation, you might think of Teenage Mutant Ninja Turtles. Or you might think of things, like the lamb born with 2 heads, or frogs with no arms, or all the other sorts of crazy things that are out there. But most mutations are not that exciting. What we're going to look at are some of the causes of mutations, the different types and effects of mutations, and finally, what types of cells mutations have to occur in order to be passed on from one generation to the next. As you may know, a lot of the different things that we are exposed to in our lives may cause mutations, and oftentimes these mutations, if they're affecting genes that are responsible for regulating the cell cycle, can lead to cancer. Some things that we can avoid in our daily lives to prevent our exposure of mutations are, of course, cigarette smoking. That is a big no-no because cigarette smoke is full of different chemicals that can cause mutations in our cells. Another thing that we want to avoid is radiation, okay? Which is why when, for example, the Fukushima nuclear reactor in Japan had a meltdown after the tsunami, that was a problem and they didn't want people living near there, so they had to move. Another thing, which is relevant to a lot of your lives as teenagers, is tanning beds. Those are another big no-no. Okay? So avoid these things. But what all of these things have in common is that they might make changes to the DNA. So that's what a mutation is. It's a permanent change to the DNA in an individual cell or in the cells of an organism. Let's look at what happens when these changes occur. This image up here kind of shows a very generalized diagram of what happens when, in this case, an ultraviolet photon from ultraviolet light from the sun or from a tanning bed causes the DNA to kind of break and a mutation occurs. But let's look at what happens in terms of protein synthesis. Remember that during transcription, right, we go from the DNA to the mRNA. Then during translation, that mRNA is coded to make the amino acids, to make the protein. A couple of things that are important to remember is that if the DNA is changed, that will change the mRNA due to complementary base pairs. Another important thing is that for mRNA, remember every 3 bases on the mRNA is a codon. Due to the combination of different nitrogen bases in the mRNA, you can end up with 64 different codons. Okay? So remember that, 64 codons. On the other hand, the proteins are all made up of different combinations of amino acids. There are 20 different amino acids. Okay? So just remember those 2 numbers because that will be important in just a moment. When we're looking at mutations, right, changes in the DNA, what we're really concerned with is how does that change affect the protein that's built. This image is showing us some different effects of types of point mutations. So a point mutation is when one nucleotide in the DNA is changed. So basically that means we're changing one nitrogen base or one letter. Okay? So over here on the left we have our example of no mutation. So we can see that the DNA normally reads TTC. So during transcription that makes mRNA AAG, which this codon codes for the amino acid lysine. Okay? And this is what the amino acid lysine looks like. Now, remember we were just saying there are 64 different codons and only 20 different amino acids. So what that means is oftentimes one amino acid has multiple codons that code for it. So if that C were changed to a T, so the DNA was TTT, the mRNA would be AAA versus AAG, the amino acid is still lysine. That's known as a silent mutation because it doesn't have any effect on the amino acid that's made or on the protein that's made. However, if we had a different type of point mutation where the first letter was changed, so rather than being TTC, it was ATC, that changes the amino acid to UAG, which you may remember is one of those stop codons. So stop codon, that would be the end of the amino acid chain. So that would really shorten that amino acid chain and could very drastically change the shape of the protein and therefore the function of the protein. Then we have different types of missense mutations, and missense mutations occur during, it's a type of point mutation where it changes one amino acid. So that one amino acid changes, in this case, to arginine or threonine, and that could change the protein. Some of these might be very minor changes, others might be more drastic. A couple of words that you may have seen before have come up in various questions maybe or discussions, or you might remember from middle school, are genotype and phenotype. So when we're talking about what's happening with the DNA on that level, that's the genotype. So what does your DNA say? And then when we talk about the protein that's actually made, okay, or the physical trait that results, that's the phenotype. So genotype, right, DNA, determines phenotype, the protein that's made. Sometimes we can have a silent mutation, so change in the genotype that does not change the phenotype, does not change the protein, and other times we can have much more drastic mutations. Here are a few more examples of those point mutations. Okay. So here's right here we have normal DNA. So our normal DNA, normal mRNA, and the normal amino acids. Here's one of those missense mutations where we've changed one letter. That changes one amino acid. Here's a nonsense mutation, so we change one letter, then it gives us a stop codon, so that really shortens it. Then we have another type of mutation down here, a frameshift. And what happens with a frameshift mutation is this is when either one additional nucleotide is either inserted, or in this case it's deleted, right? So this nucleotide that had an adenine gets kicked out, and what that does is it changes the codons, right? So AUG, AAG, those stay the same. UU, see back here it used to be UUU, now it's UUG, and then GCU, then AA, so on. So it changes everything after the mutation. So here's our mutation point right here. Okay. All of those codons now, as you keep going, every 3 bases will change. So that's a frameshift mutation. Those can be much more drastic than a point mutation because it's going to change all of the amino acids further down that gene. Here we have another representation of additional types of mutations. I was just talking about before a deletion, which you could have, right? So when you delete something, in this case it might be a whole region of a chromosome, so multiple genes or part of a gene versus just one nucleotide. You could have a duplication, so in this case we have 2 Bs, right? Here B is missing. Now we have duplication, so an additional gene or multiple genes that have been added in. You could have an inversion, so rather than reading A, B, C, D, E, F, this whole part has been flipped around, right? It's flipped around. That could change things because where a gene is located on a chromosome affects how often it's going to be transcribed and then translated to make proteins. You can also have a translocation where parts of genes switch places. So we're getting kind of a flip-flop here. Let's look at these with some actual chromosomes so you can get a better idea of what's happening. Up here on the top, we can see different single chromosome mutations. So the deletion, so we're missing a part right here. Duplication, when you end up with an extra part. Or an inversion, kind of hard to see, but if you look at the banding pattern on these 2 chromosomes, it's been flipped around. So those are mutations that can occur on one chromosome. Okay. And then down here, we have mutations that involve multiple chromosomes. In an insertion, you actually still have all of the genetic material in the individual cells, but part of it, so in this case, part of chromosome 4 ends up in chromosome 20. That can change things when you think about like meiosis and how those chromosomes line up, and can again affect how proteins are made. And then we also could have a translocation. In this case, chromosome 4 and 20 have actually flipped parts. This is different from crossing over because crossing over would occur, for example, between the maternal and paternal homologue of chromosome 20, and they just exchange information, but the same genes. In this case, genes, gene locations have changed. So again, that can change things. When we start talking about genetic disorders later on this year, after mid-year exams, we will be talking about some of these different types of mutations and what effects those can have on individuals. And we'll be focusing mostly on human genetics. At this point, it's very important to point out that in order for a mutation to be passed on, it has to occur in specific cells. So remember, we all started off, right, as an egg cell and a sperm cell will fertilize to form a zygote. Okay. Now, the zygote goes through mitosis and develops, right? So you have somatic cells, all of your body cells that are making up most of your body, and then the germ cells. Germ cells are the ones that go through meiosis to become egg or sperm. Now, if a mutation occurs in any of your somatic cells, that mutation ends with you. It cannot be passed on to your offspring. However, if a mutation occurs in a germ cell, that mutation could end up in sperm or egg and therefore affect the next generation. So mutations must occur in the germ or sex cells, okay, in order to be passed on. And that's important to keep in mind. And then oftentimes, when we're thinking about genetic disorders, a lot of times those are not resulting from mutations that were passed on, but they're from spontaneous mutations that occurred maybe during meiosis or even very early on in development. So we'll be looking at all of that in about another month.